Hello, my name is David Earl Hostallero, a PhD student at McGill University supervised by Professor Amin Ahmad, and I will be presenting our research in prediction of cancer drug response using heterogeneous graph convolutional networks. Lately, studies have framed drug response prediction as a paired input prediction problem, where a drug and a subject constitutes the model's input. This approach induces information sharing across drugs and subjects compared to drug-specific models shown below, which are inherently independent. Many models use the structural properties of the drugs as their representation, as this information is easily available. However, effects may still vary across drugs even with similar structures. For example, on the right, we have two groups of cell lines that are sensitive to different group of drugs. It makes sense that even if the drugs within each group may have structural differences, they should have similar embeddings because they are semantically similar. On the other hand, if the top left drug group has an opposite effect to the bottom right cell lines, drugs from different groups should have low embedding similarities. How do we do that? We use information propagation using graph neural networks. We developed two models, called BigDRP and BigDRP Plus, that use a bipartite graph containing drug and cancer cell line nodes. BigDRP utilizes HGCNs, or heterogeneous graph convolutional networks, to propagate information from drugs to cell lines and the other way around, therefore incorporating some cell line information into the drug representation. The model uses three types of data: the drug response matrix in the form of log IC50 of the training set, the gene expression of cell lines, and the drug descriptors. Using the log IC50 of the training set, we filter the top and bottom k% of responses for each drug corresponding to the most resistant and sensitive cell lines. By default, we use k is equals to 1. After filtering, you would see that this filtered matrix can be translated into an adjacency matrix of a bipartite graph with two types of edges. Each node is either a cell line or a drug. Drug nodes use their drug descriptors as attributes, while the cell line nodes use their gene expression profiles. This heterogeneous bipartite graph is then fed to a two-layer heterogeneous graph convolutional network, or HGCN, which will produce node embeddings. The HGCN allows us to incorporate the semantic similarity that we have presented. We also added an external CCL encoder FC, which allows us to predict for new cancer cell lines. Due to the data splitting and filtering, it is possible that some cell lines are not part of the bipartite graph. Therefore, this external module is needed. The BigDRP Plus is a fine-tuning of the previous model that allows the predictor to learn without the HGCN's message passing, therefore stabilizing the predictor by removing all the elements that may cause the drug embeddings to change unnecessarily. We compared our model to multiple baselines and competing approaches in different cross-validation splitting schemes. The leaf pairs out LPOCV and the leaf cell lines out LCOCV. The LPOCV assesses the model's ability to fill experimental gaps, while the LCOCV evaluates the model's generalizability to unseen subjects. Both BigDRP and BigDRP Plus outperform other approaches and baselines in both schemes. We would also like to note that these performance improvements are statistically significant using a one-sided Wilcoxon signed rank test of the Spearman correlation coefficients. This is also true for BigDRP Plus and BigDRP, albeit a small amount of per drug improvement. Our evaluation on different values of k shows that performance is also stable with a slight deterioration as k increases. We were also curious about the structure of our bipartite graph and found that there are 5 drug clusters. Comparing each cluster's performance against a baseline MLP, we found that all clusters have shown a significant improvement, with cluster 3 in green having the highest median improvement. In our analysis, we found that cluster 3 is mostly composed of protein kinase inhibitors. This suggests that some of the drugs in the same cluster have similar mechanism of action that can be captured by our bipartite graph. In the future, we plan to investigate these mechanisms of action. We also evaluated the mutation landscape of tumors and their association with drug response. To do this, we predicted the response of TCGA tumors for the 237 drugs that we trained for. Tumors with PIK3CA mutations are significantly more sensitive to drugs targeting the PI3K, AKT, mTOR pathways. Conversely, tumors with PIK3CA mutations were associated with resistance to drugs targeting the MAPK-ARK signaling pathway. Both results are consistent with prior studies. Additionally, thyroid carcinoma tumors harboring BRAF mutation are significantly more sensitive to drugs targeting MAPK-ARK pathway. Prior studies have demonstrated similar results, such as Wang and colleagues in their paper referenced below. In summary, we presented drug response prediction models, BigDRP and BigDRP+, which create drug representations through the propagation of drug and cell line information using graph convolutions. Our models surpass baselines and other competing models in different data splitting scenarios. The bipartite graph could provide similarities beyond the molecular structure or properties of the drug. And our evaluation of the mutation landscapes of tumors are consistent with existing literatures. If you want to learn more, please visit us in the poster session or read the paper on the right. Thank you.